All right, so now we're going to begin our discussion on cellular levels of organization, tissue level of organization, as well as cellular transport. So, really, one of the first things we are going to talk about is the cell membrane. The cell membrane is a phospholipid bilayer, meaning it is two layers of a phospholipid containing substance. You'll see on the screen right now a diagram, an illustrated diagram of what this means. We essentially have a head-like structure and a tail-like structure. The head is going to be made of a phosphate group combined with a glycerol molecule, and those will be bound to the two tails, which will be fatty acids. These two different molecules will be in place on the outside of a cell, similar to how I'm drawing right there. So you will see that the tails face inward and the heads face outward. This is important because the head and tails have a favorability for water. Now, the heads are hydrophilic, meaning they are okay being exposed to water. And beyond water, we're also talking about things that have charges, okay? The tails are hydrophobic. These things do not like being exposed to water or other charged particles, okay? They are, fearful of being exposed to those things. Now, this is important for the function of the cell membrane because the membrane is selectively permeable. Okay, this means that certain particles have the ability to go through the membrane without requiring any energy, without even requiring membrane proteins, which we will talk about shortly. And the key here is that the hydrophobic tails do not like being exposed to those charged polar particles. So if we have particles that aren't really polar, things like other lipids, they can actually go right through the cell membrane without any special transport proteins, without spending any energy, etc. Now, if we're talking about things that do have charges, that's going to be water. It's going to be electrolytes, so things like sodium, potassium, chloride, calcium. Those are really four really big ones, and magnesium as well, which is often neglected but super important for cellular function. Those all have charges, right? Those are all salts. Salts are notoriously charged. Those things will require some form of help to get through the membrane. And that is where these membrane proteins come into play. These proteins can be completely transmembrane, meaning they go through the full membrane itself, or they only are on one side of the membrane. Now, there are a couple general classes with them, and a couple ways we talk about them. There are integral proteins and peripheral proteins to start off, but integral proteins are these transmembrane proteins that are fully integrated through the membrane. All right, one major type of these are channel proteins. These channel proteins have a very specific shape and a very specific charge, and this allows very specific substances to flow through them. A common example of this could be something like a sodium leak channel, and it's called a leak channel because the inside of the cell will contain more sodium, and the sodium is able to essentially just flow out of that channel. And again, This is a type of passive transport, and we will be talking about transport types in a little bit later. But these proteins can do things related to transport, especially something like a channel protein. Next, we have the peripheral proteins. These tend to be the ones that are found either on the outside or the inside, but do not go fully through. Now, they often are associated with other proteins since they do not fully extend through the cell membrane. And so we can see something like a receptor being paired with a peripheral membrane protein. And receptors are something that we will see on the membrane that specific molecules will bind to. Okay, so for instance, you could have a hormone that wants to bind to the cell to signal something to happen within the cell. So the hormone doesn't have to actually go inside of the cell to make this happen. It will bind the receptor on the surface, and that receptor can activate a peripheral protein that would then send a signal into the cell, and that's called a second messenger system at that point. One of the most common second messengers is a particle called cyclic AMP, often abbreviated CAMP. Another item you will see labeled here is glycoproteins. Glycoproteins are very important in cell recognition, is one of the things that we will talk about in this course. And the reason cell recognition is so important is that it's one of the processes in which our white blood cells actually can sort of look at a cell and say, hey, you're supposed to be here, you're part of us, you're on my side, right? Because the white blood cell exists to fight off non-self material, destroy anything that potentially can damage the body, anything that's, a bacteria, a virus, etc. If it doesn't have these, essentially if a object is big enough, it can initiate an immune response if it doesn't have certain properties, okay? And the presence of a glycoprotein on a cell membrane is important for that, okay? And that's another thing when we talk about the immune system that will come up is, especially with bacteria, right? The human body, we have a cell type that's called eukaryotic cells, and eukaryotic cells do not have cell walls. We have a nucleus that's contained within a nuclear envelope, etc. Whereas prokaryotic cells, like bacteria, many of them have some form of cell wall. They don't have an organized nucleus, etc. And the proteins on the outer surface of these prokaryotic cells are very different than the ones on our cells. Okay, so these proteins are important in cellular recognition, okay, and are a major part of our immune response. Now, moving into transport. So transport really comes in two dimensions. It's either passive or active. And the easiest way to separate these two is that passive does not require energy. It does not require ATP. Okay, remember ATP is the basic energy molecule used by our body. All right, everything that is used for energy gets converted to ATP for it to actually be used. ATP being adenosine triphosphate. Active transport often requires the help of, things like proteins, and those proteins may need ATP to function properly, okay? And we'll talk about a specific example, the very classic example of the sodium-potassium pump here shortly. But this brings us back to our idea of the cell membrane being selectively permeable, okay? So again, remember the heads of the phospholipid bilayer that face out of the cell and into the intracellular environment, they are hydrophilic. They are okay being exposed to water and do not react with water. Whereas the fatty acid tails that are on the inside of this membrane, they face each other. They don't need to be exposed to these polar charged molecules. They don't like it. They don't biochemically get along with those molecules. And this is where the idea of selective permeability comes from. Again, remember, very small molecules, non-charged molecules, lipids, gases. These things can be pushed through without any sort of energy expenditure required, right? And even some things like leak channels, there can be some very minor passing of these charged particles, but they are using a protein. So again, it's a little different, okay? Now, passive transport. The big thing to think about this is that materials in the body form a concentration gradient. This means that there will be substances inside of a cell and substances outside of a cell, but they can have different concentrations depending on which side of the cell membrane you're on. And following basic physics and biochemistry properties, these molecules will want to actually spread themselves out evenly. So if we have a ton of sodium inside of the cell and we have very little potassium outside, or we have very little sodium outside of the cell, naturally those things would want to even out to where it's eventually equal inside versus outside. And this movement that happens naturally through the absolute basic properties of physics and biochemistry is called diffusion. Okay, so these substances will move down a concentration gradient from high concentration to low. Now, there is something called facilitated diffusion, and I hinted at this earlier talking about the leak channels. We can have facilitated diffusion where a protein or some other molecule assists a large or polar charged molecule to go through. We can also have dedicated carrier proteins that exist for these as well, like how glucose is transported. You'll see examples here in the lecture. They are basically showing you the examples of carrier proteins, channel proteins, and osmosis. And osmosis is literally the same exact concept, but with water. And when you look at the example here, it shows us how we can actually get water to follow other molecules, okay? And this becomes important when we talk about things like the kidney, where salt balance is actually crucial for excretion of excess fluid. And that's why in the example picture that you see here in the lecture, we can actually, within that beaker, make the water high on one side. You would think that that couldn't happen, but it does with the ability of water to follow these other molecules. Now, another important thing to consider with osmosis is whether a solution is hyper or hypotonic. So hypertonic solutions essentially mean that there's a higher concentration of non-water molecules outside of the cell. And there is less water, there is less water molecules outside of the cell. What happens here is to try to even this out. The water inside the cell is pulled out. So these cells will actually shrivel up. In an isotonic solution, there's a relative good balance of water in and out of the cell. And this will cause, this is ideal, right? You have water go in, water go out, and it's in an even amount going in and an even amount going out. The cells are the correct size, etc. Now, in a hypotonic solution, the external environment has a lot more water and it doesn't have a lot of substances like salts. And this will cause the water to get pushed into the cell. This will cause the cells to expand, and then eventually they will lyse open, okay? So, one last bit of passive transport to talk about is hydrostatic gradients. This is essentially pressure gradients that are created by the flow of fluid. This is something that's can be important for capillary blood flow. It's kind of the end product of your blood pressure at the capillary level, where the nutrients and oxygen that our blood carries gets pushed out of the bloodstream. And the same thing is also very important in the kidneys, because blood pressure at the kidney also is used to push fluid through the nephron, which is the filtering apparatus of the kidney. Now, active transport, again, active transport requires ATP. It requires energy, and often this is because a protein will be moving a substance against the concentration gradient, okay? So we need to move things in a different direction than where they would naturally go. The classic example of this is the sodium-potassium pump. So with this sodium-potassium pump, if we have a lot of sodium in the extracellular space and a lot of potassium in the intracellular space, this becomes important because there's a charge differential across the cell membrane with these substances being present. And we'll dive more into this in future weeks when we really talk about muscle physiology and neuron physiology. But those two cells really rely on this charge differential to do things like contract, send action potentials, which are nerve impulses. And to get a nerve, for instance, to send an impulse, we have to alter the charge across the membrane. And by doing that, we use the sodium-potassium pump. Okay, so ATP will essentially bind the pump, which then allows the protein to change its shape and push, the sodium or the potassium through. For every cycle of the pump, there are 3 sodium ions kicked out into the extracellular space and 2 potassium ions brought into the cell. Okay? That's very, very, very important. Now, other forms of active transport are a little different because they don't necessarily involve membrane proteins. Now, the first of these is what's called phagocytosis. This is cellular eating, okay? So this is for large solid particles. This is something commonly associated with immune cells. So our white blood cells love to do this, especially our T cells and our neutrophils, right? So these guys, they find a substance that, isn't identified as us. So remember, let's just say there's a bacteria floating along in the bloodstream and it doesn't have that glycoprotein on it that says, hey, I belong here. So the white blood cell essentially targets it and eats it, quite literally. So what happens is using some of the proteins within the cell wall that we'll be talking about here shortly, it bends its cell membrane around that substance, pinches itself off, and forms what's called a vacuole. Now, these vacuoles can take many different forms, but in a white blood cell, the substance gets pulled in and it's put inside of something called essentially a specialized vacuole that's a lysosome. And as the name implies, it causes lysis of whatever is in it. It breaks it down, rips it apart. It essentially uses chemicals that are like acid, okay? Then we have something called pinocytosis, and this can be thought of as cellular drinking. Cells can bring in large amounts of external fluid and things like electrolytes or other molecules dissolved within that fluid rather quickly via this method, and it essentially follows the same process. The cell membrane will invaginate and cut itself off and form a vesicle that will then eventually be emptied. Now, receptor-mediated endocytosis, this essentially can happen for a couple different reasons. Some immune cells actually do this. B lymphocytes will do this, but macrophages will do this. Macrophages, as the name says, phage means to eat. So macrophages love to eat particles and then give the signaling particles that say, hey, this thing I just ate, it shouldn't be here. Somebody help me get rid of, the rest of it. And it then gives that particle to B cells, T cells, and other white blood cells. So receptor-mediated endocytosis is kind of what the macrophage does there, right? It binds a protein and then it essentially eats it, it endocytoses it, and it will digest it inside of itself. But we also see this happening with things like proteins, energy substrates, etc. They bind a receptor on the outside, and that whole receptor ligand complex is brought inside of the cell. And that's one of the big differences between that and general phagocytosis. On the opposite of that, though, we have exocytosis. And this is how a cell will get rid of things like packaged proteins. And we'll talk about how that happens here shortly. But proteins, other molecules, even waste, it will package it up in a vesicle. That vesicle will then bind the membrane, and then it's expelled. Very simple, almost the exact opposite, okay? And we're now moving on to organelles. So the first big thing to think about when talking about a cell is that there's essentially various parts of the cell, and as the name implies, organelles just means little organ, small organ. They really are the organs of the actual cell. The cell can't function without many of these, and these things don't really do anything without being in the cell. Now, the first thing to really think about is the cytosol or cytoplasm, and this is the gel-like substance, mostly made of water, that fills the empty space inside of a cell. Everything is within the cytosol, and there are some important things that actually happen in the cytosol, like a lot of our anaerobic metabolism happens there. Glycolysis, for instance. Now, the first main thing to talk about is the endoplasmic reticulum. We really have two organelles that function to transport and package substances, and the first is the ER. Now, there are two types of ER. There's a rough ER and smooth ER. The rough ER is named rough because it has ribosomes bound to its surface. And ribosomes are the organelle in which protein is created and sent out from, which makes a lot of sense if we need to package it and send it elsewhere. Being attached to the endoplasmic reticulum is good. Now, the smooth ER is associated with a lot of lipids and fat creations. It's also important for regulating the concentration of calcium, and this becomes important when we talk about muscle. And in the muscle, it's specifically called the sarcoplasmic reticulum, and that calcium role is much, much greater there because calcium really is the key. On top of ATP, calcium really is the key substance to cause muscle contraction to occur. Both of these, they package these substances and they send them to various parts of the cell. Now, the Golgi apparatus, on the other hand, can kind of be thought of like the post office. It modifies and ships products. These products often come from the endoplasmic reticulum. Now, the Golgi apparatus also can package these things into vesicles, and these vesicles can have exocytosis occur to them. So this could be a cell, for instance, a cell in the substantia nigra of the brain creating a neurotransmitter, a substance called dopamine. And it could create this product and throw it out into the extracellular space for it to go have its effect. The Golgi apparatus also does stuff with digestive enzymes, etc. as well. Now next, the mitochondria. This is where the majority of our energy is made in the body. We do have a little bit made in the cytoplasm, primarily through anaerobic means, but anaerobic means are very inefficient for us. We are anaerobic organisms. And as such, we need oxygen. And this is really where all of the oxygen that we breathe really needs to end up. Okay, some of it is used for other random things throughout the body, but a majority of it is necessary for our ability to produce energy. And it's a process called oxidative phosphorylation, which we will talk a little bit more about later, but it essentially allows us to take the products of multiple different energy substrates and turn them into ATP in a very, very, very efficient manner. For instance, a normal glycolysis pathway nets us only 2 ATP. That is enough for only one very, very, very small part of one muscle fiber to contract once. Okay? Whereas glycolysis nets us well over, oxidative phosphorylation nets us well over 30 ATP per cycle. Okay, so there's a major difference in efficiency. You'll see lysosomes and peroxisomes mentioned. We've pretty much already talked about these. They're involved in metabolism and breaking down other substances. Now, the cytoskeleton. I mentioned how in like phagocytosis, the membrane can become distorted and wrap itself around different things. And part of that is the cytoskeleton proteins. There's 3 main proteins, the first of which is the microtubule, which is primarily formed of the aptly named protein tubulin. Microtubules really function for shape and structure and stop the cell from being squished. They also play a major role in some structures that come off of the cell membrane, specifically cilia and flagella. Cilia are a structure that can kind of be thought of like arms or hands. They often move rhythmically, and one place we see them is in the respiratory tract. And this is because the respiratory tract has a lot of mucus. And that mucus needs to constantly be moved through the respiratory tract and brought up. This is generically called the mucociliary escalator. But believe it or not, this mucus plays a major role in our body's immune system. It catches a lot of stuff and stops it from getting into the lungs. The cilia is involved in that by pushing it out. Okay. On the other hand, flagella can be thought of as kind of like the equivalent of the tail of a fish on the outside of a cell. Now, we as humans only have one cell with a flagellum, and that is the sperm. Now, there is some complex interactions here, but our body, since we only have those one cells, our body actually, the immune system will be triggered on flagella. So many bacterium have flagella. And they're essentially made of the same exact structure as in our bodies. And since flagella are so rare, it's one of the reasons why sperm cells are actually kind of sequestered from the immune system, because they will actually trigger an autoimmune response. Then we have something called the centrioles. Centrioles are very important in the processes associated with DNA separation during the cell division as part of the cell cycle. And then we have microfilaments, and these are pretty small and intermediate filaments, which again, they're intermediate and are important for shape, structure, just like all the rest of these. Now, the nucleus can be thought of as kind of the brain of the cell. It's also the location that our DNA is kept. Surrounding the nucleus, we have what's called the nuclear envelope, and this kind of helps protect the nucleus from anything that does manage to get into the cell. It's kind of a last barrier layer. Now, most cells in the body only have one nucleus, but it is possible for cells to have multiple nuclei. And a prime example of that would be skeletal muscle, for instance. It's very, very common to see skeletal muscle with multiple nuclei. And this is because skeletal muscle are so reliant on the production of protein, and protein really does come from the DNA stored within the nucleus, so it needs a large amount of protein to be produced at all times. Now, moving on to the cell cycle and DNA replication. So, a few things to really talk about. Cell replication. Most cells in the body do replicate relatively regularly. Some of them do not. And some of them, rather than replicate, they undergo just generalized renewal of themselves. That would include something like nerve cells. Now, it is possible for nerve cells to divide. It is incredibly rare and not reliable for any practical means currently. We can make it happen, but only in the laboratory environment. And the same thing can be said of skeletal muscle and cardiac muscle that it's mentioned in here, but we've had no success doing it in a living person. That does not mean, though, that these cells cannot repair themselves, especially when you talk about nerve cells and muscle. Those things are actually, especially peripheral nerves as well, they're relatively robust and can repair themselves provided the damage is not too extreme. Now, basic structure of DNA, okay? It's a double helix, looks like a twisting ladder, okay? The double helix is really two separate strands of complementary DNA. DNA will have three basic molecules. There's going to be a sugar, a phosphate, and then a nucleic acid. And we have 4 nucleic acid bases, okay? These are adenine, thymine, cytosine, and guanine. As you see, they are all abbreviated by their first letter within their name. And they always pair with a specific other molecule. So A and T always go together. C and G always go together. Okay? They will always be bound that way. All right? They have complementary, as we'll see in another picture, they have complementary bond formation, their shapes match, etc. They fit each other very well at the biochemical level. Now, what is really interesting about DNA in the body is that at really the top level, we have the chromosome. In the chromosome, we have 46 total, 23 pairs, and all of our DNA is stored across the 23 different chromosomal pairs. It's tightly wound together, as this shows us, into what we call chromatin. And chromatin really consists of all of the tightly packed DNA. And this DNA will actually wrap around another protein called a histone, as you can see illustrated here. And the histone that has DNA wrapped around it is called the nucleosome. All right. So we can, despite our DNA being able to literally, if it was end to end, it could wrap around the earth multiple times. Yet it's packed into this microscopic cell. It's because of these histones. These histones are key for that to happen. But for DNA replication to occur, we have to unwind all of this. So we have special proteins that exist to unwind the nucleosome, to unwind that. And then we have another protein that kind of unzips the helix. So as you see the bottom of this diagram here, it's unzipped. Once it's unzipped, we have a process begin where either the DNA is copied in a complementary manner, meaning you will then create at the same time 2 strands that are still identical to the original, but then they will bind together. Now, what typically happens with our DNA is we have a mixed division where one of the new strands binds to the old strand that is complementary of it. Okay. Now, once that's done, we can also see processes where DNA is turned into a substance called messenger RNA. Now, RNA also uses nucleic acids. It is in humans, though, our RNA is all single-stranded. And we really look at the RNA in a codon or triplet. And when the RNA is created, we're going to see the nucleic acids in sets of 3. And those actually are essentially the code to tell the ribosome to take and find an amino acid. So the amino acids are kind of laid out and then attached, and that's how the protein is formed. So messenger RNA goes from the nucleus to the ribosome. And really, this is the process that's called transcription. Translation occurs at the ribosome, where this messenger RNA is fed in, and almost like an assembly line, as it rolls through, the ribosome translates it, and the aptly named translation, into a protein. So it pulls all the amino acids in, and it does that with the help of tRNA, transfer RNA, will bind the necessary amino acid and drag it into the ribosome if it's not already present. And that's essentially the basics of how a protein is formed, because you'll eventually hit the end of the messenger RNA and the protein will be released. And if it's from a ribosome on the rough endoplasmic reticulum, it gets packaged and sent to its final destination. Now, cell cycle. Big thing to think about the cell cycle. We essentially have two phases. All right, there's the interphase and there's the division. Division is really divided into two things, which is mitosis and cytokinesis. Mitosis is when we divide genetic material and replicate it. Cytokinesis follows up mitosis so that then the cell literally will split itself in two, and we'll have two fully functioning cells with all of the necessary genetic material in each of them. So with interphase, this is where the cell is not actively undergoing any division. We really just have growth phases or gap phases and synthesis phases. All right, and these really don't matter all that much for what we're going to be talking about. Now, for actual mitosis, mitosis really is 5 phases. So with prophase, we really see the cell start to prepare, right? The chromosomes start to do what's called condensing, and the spindles will start to form. With metaphase, our chromosomes kind of line up and chromatids will attach, so that in anaphase, the next step, the chromosomes essentially ripped in two, okay? And again, this is the chromatids. Once that happens, the nuclear envelope reforms around them, and then cytokinesis occurs where the membrane invaginates and we break into two cells. Now, next, tissue organization, okay? We have 4 main types of tissue in the body. There's nerve tissue, epithelial tissue, muscle tissue, and connective tissue. There's many different types of cells underneath all of those, but these are the 4 generalized tissues. Nervous tissue, makes up the brain, the spinal cord, and nerves. We have muscle fibers. There's 3 main types of muscles. There's cardiac, there's smooth, and there's skeletal. We have connective tissue. There's many different types of connective tissue, but this is going to include things like the bones, your tendons, even your blood is a connective tissue. Fat is also a connective tissue. Then epithelial tissue can be thought of as liners, right? Epithelium is the skin. It's what lines hollow organs, okay? So it's kind of the lining tissue. So epithelial tissue, it's often closely packed. There's very little extracellular matrix, but most epithelial have a basement layer to separate it from the underlying tissue. Epithelia primarily functions as some form of barrier and is associated, really with lining things, right? They form what's called tight junctions often, which literally prevents almost anything from passing between the individual cells. Now, when we talk about epithelia, we really have a couple classifications and a couple of arrangements. There's literally 2 arrangements. We have what's called simple epithelia and stratified epithelia. Simple epithelia is just that. It's simple. There's one layer of cells. Stratified means there are multiple layers of cells. Okay, so all of these epithelium we're going to talk about here shortly can come in simple or stratified. We also have pseudo-stratified, but that is relatively rare and occurs because the cells are oriented slightly differently from each other. So while there's only one layer, when you look at it on a microscope, it actually looks uneven, okay? But it is one layer. So, first thing to talk about is squamous epithelia. The squamous epithelia, they're flat, all right? They're very flat. There's not much more to them than that. Simple epithelia makes up, squamous epithelium is a big part of our skin. Then we have cuboidal epithelium. These are literally cube-shaped cells. We see a lot of these in the kidney. They also line glandular ducts, etc. The nucleus for these guys is also very close to the middle of the cell. They're associated really with secretory function and absorption as well, which makes them really good for the kidney and for glands. Columnar epithelia, they're tall. They look like columns, right? The nucleus sits towards the basal layer, and this is part of how we can end up with the pseudostratified columnar, because the nucleus in that type doesn't sit next to the basal layer, so it actually can make it tricky and make it look like there's multiple different cells when only there's one, okay? We see a lot of columnar epithelia in places like the intestines, in the digestive system, in reproductive tracts, etc. Pseudostratified columnar epithelia, these guys hang out in very few places. One of them is the bladder, for instance. But again, they're columnar and they look like there's multiple layers, but there's only one. So this is a labeled description of each of them. We won't dive into that. It's also important to know that certain cells, like columnar epithelia, are prone to have cilia. So, connective tissue. First thing to think about connective tissue is that it's structural, okay? So, it also makes up, it's one of the most, it is the most abundant tissue type in the body. It fills all of the extracellular space. The major cell type involved in this is a fibroblast. These guys are the primary cells that build. So when you hear the word blast, you think build, okay? These guys build connective tissue. Another thing to consider is the two different osteocytes, the osteoclast and osteoblast. Those guys are connective tissue. And hematopoietic cells, so red blood cells, white blood cells, leukocytes, lymphocytes, all of those guys are all connective tissue. Now, we have different types of connective tissue, but we don't really have to get too deep into it. Loose connective tissue is just that, it's loose. It's going to make up stuff like adipose tissue, random tissue that fills space in the body. And then we have dense connective tissue. Dense connective tissue makes up things like tendons and ligaments. Just as it says, it's very dense, it's rigid. We have cartilage, which is made by chondrocytes. And, the cartilage, it lines joints. There's also various different types of cartilage. And it's primarily made of collagen, but cartilage lines joints. It helps provide, the basis for things like the ear, the external ear, the nose, etc. Muscle tissue. We won't get too much into muscle tissue now, but in general, there's three types. Skeletal muscle is the only type that is voluntary, meaning it's the only one that we can consciously control. Cardiac and smooth are involuntary. Skeletal muscle and cardiac muscle are what's called striated. So when you look at them under a microscope, they have lines through them, and this is because of the way they are structured. They have various pieces to them that allow them to connect in a straight line. Smooth muscle does not have striations, okay? And this is because the proteins within them are not aligned, all right? They're a little more random, but not as random as you would think, and it's because smooth muscle doesn't really contract in a straight line. It contracts more of a elongating type way. Now, nervous tissue. The neuron is the primary nervous cell, but there are others. There's things like the astrocytes, microglia, Schwann cells. You often see Schwann cells and oligodendrocytes. Both of those guys actually provide myelination of the neuron, but all the guys I just mentioned, they are really support for the neuron itself. The neuron has a large body with multiple dendrites coming off of it. You can think of the dendrites as the place where the neuron, we call it synapse, with other neurons. And the other neuron will synapse to it via what's called the axon. And the axon is the, not necessarily single, but there can be multiple axons. And it will extend, and the axon is where signals are actually passed down. So an action potential occurs, and it is sent. Within neurons as well, the area where the nucleus is contained, we call that the cell body or the soma. Soma is often the term associated with the cell body and neurons. Now, talking about some of the support cells, oligodendrocytes and Schwann cells, they actually will wrap themselves around the axon and essentially act as an insulator. And this is important because the neurons that have that, they're termed myelinated. This actually speeds up transmission and is very, very important. We have some diseases in the body that cause demyelination, and it results in a lot of issues. Things like multiple sclerosis, MS, or ALS as well. Those things are demyelinating diseases, and they have widespread prominent issues. Astrocytes and microglia, those are other support cells involved in stuff like blood-brain barrier, important as white blood cell type function within the brain, etc. So that is everything for the week 2 lecture.